Hello, welcome to AP Biology or College Biology, Unit 3, Cellular Energetics. We're focusing in on Section 3.6, which is all about cellular respiration. Please note the disclaimer listed below, and let's get started. Now, before we go into the nitty-gritty that is associated with cellular respiration, I want to preface this video by saying that this video is really broken down into several components, including the brief overview of cellular respiration, and then we go into understanding the difference between the different steps in cellular respiration, and then going into the understanding and differences between aerobic and anaerobic respiration. This section in general is very difficult because there's a lot of information that you don't know what you need to know for the exam. So I want to preface this by saying there's a lot of details in the information given to you in this video. And that's because I'm trying to give you as much information. So if you get, say, a really hard free response question where they give you a step that is more biochemistry, you can piece it together and achieve the 4 or the 5 that you deserve because you know more details than what's actually given to you in, say, a overview for a AP biology book or college biology book. So I want to preface this by saying that this video is going through basic steps, then more detailed steps, and then tying it all together. If you want more specific information about specific steps, make sure you scroll down below and hit this appropriate timestamps so you can review the section that you want to review that you might need for an exam or test. Also note, if you're trying to get review questions, there is a question at the end of this video, so make sure you stick around for that. Okay, let's get back to this video. So remember, in Unit 2, or maybe in a previous section, if you're in college biology, you've spoken about the mitochondria. And so for cellular respiration, the mitochondria is a very important organelle. Just to recap, remember, mitochondria has all these parts, and you covered this usually in a previous unit around Unit 2, and we're going to recap those parts. Remember, there is an outer membrane for the mitochondria. And that outer membrane is the initial protection for the mitochondria. Remember, the mitochondria is located, if you have a picture of a cell, you have the nucleus. The mitochondria is usually an organelle that is outside the nucleus, but inside the cell. So it's usually somewhere in the vicinity of the cell in the cytosol. So the mitochondria itself, if you remember in previous units, we talked about the endosymbiotic theory, which basically states that the mitochondria has actually evolved from a previous prokaryote. And this is important to note because the mitochondria itself is an entity in of itself. It is thought to be very evolved and in fact produces its own DNA. So that's how important the mitochondria is, and it's even more important when discussing cellular respiration. It's important because we know that the mitochondria itself is utilized to produce ATP, which we consider to be energy. And for the cellular respiration process, ATP is set to be produced mostly not only in the first step, but in the second and third step, where mitochondria is very important. And those two steps are the Krebs and the electron transfer chain. So just to reiterate, mitochondria utilizes Krebs cycle and the electron transfer chain, which are two very important steps for cellular respiration. We'll go into more detail about those in a later part of this video, but just for now, you need to understand that you produce a lot of ATP for cellular respiration in general, and it is produced in the latter steps of cellular respiration, which is the Krebs cycle and the electron transfer chain. Now, just to recap, remember, we have two membranes for mitochondria. We have that outer membrane and that inner membrane, allowing it to be a double membrane. We also have a highly folded internal portion, which formulates this matrix and is formulated with these little folds called cristae, and together they work to increase the surface area within the mitochondria, which is great because we need the greater surface area for mitochondria because it allows us to produce ATP, which is very important because efficiency to produce ATP means efficiency to produce energy itself. Let's talk about a brief overview of what we're going to see in this video, but let's integrate it with previous units to get a brief overview of what we're talking about. So if you remember in Unit 1, and if you haven't covered Unit 1, by the way, you might want to head over to the playlist related to Unit 1 where you can cover all the material related to different macromolecules that you need to understand for later units. And so when it comes to Unit 3, which is cellular respiration, remember there are different macromolecules like carbs and fats and proteins, and these macromolecules work together in our food sources. So they are built in our food sources, and we consume them as human beings to be fuel. The way that it is fuel is because these food sources provide us essentially with sugar or glucose. And if you remember from Unit 1, that's the same thing as saying C6H12O6. And to recap, oxygen is also something that we breathe in as well. So we breathe in oxygen and we consume foods. And by doing this in cellular respiration, we release carbon dioxide. We also release some water, so we release some H2O, and we also go ahead and release some energy as well. So that we say is ATP or heat. Now, this all occurs in different parts of the cell, but the majority of cellular respiration occurs in the mitochondria. So, when we're talking about the different steps, we're talking about the fact that we have what's called the cytosol. And the cytosol, if you remember, if you're looking at a picture of the cell, that's the outside membrane that is more like the fluid-like structure, not outside structure, but I should say in the cell, there's fluid jelly-like substance that we call the cytosol. It's the inner membrane space, you might want to call it, and that cytosol, which is the area listed here, holds different organelles like the mitochondria and the nucleus, and all of these organelles are within the cell. If you want more details about the specific organelles, you can follow the playlist related to Unit 2, which is all about cells and their organelles. And it will show you that the cytosol, which is in the space here within the cell, holds the mitochondria. And this first takes into account the first initial step of cellular respiration, which is called glycolysis. And it takes what's called glucose or the sugars that you consume and turns it to pyruvic acid. And we're going to go into more details into this, but just understand that the purpose of this is to produce a very small amount of ATP, which you're going to cover later on. There is also another intermediate step which occurs that produces some NADPH, and also remember we have this ATP that's being produced. Well, now the next step from glycolysis is called the Krebs cycle, and once again we'll cover more details into that. But for now, you just need to understand that that occurs in the mitochondria, and now we also have some production of ATP as well, and NADH and FADH2. Essentially, the production of these items from glycolysis and Krebs work together to then churn out electron transport chain, which is the last and final step. In this step, what happens is you have the NADH and you have the FADH2 and the NADH that's produced from the previous steps to then run the electron transfer chain, which in all of this is going to go ahead and release the appropriate amount of energy that is required for us to continue to survive. Now, when we're talking about cellular respiration in general, we're going to cover two aspects of respiration in general, and that's the difference between aerobic and anaerobic respiration. But you are expected to understand that cellular respiration, the reaction itself, is the opposite of photosynthesis, which was the previous section that we covered. Remember, in photosynthesis, we're taking carbon dioxide and we're also taking water and the energy from the sun to then produce the sugars and the oxygen that is required for us to go ahead and also conduct cellular respiration. So for photosynthesis, it's going in this direction, but for cellular respiration, we're going in this direction, where we're going ahead and saying we have our sugars and we have our oxygen, and they're going to go in to us humans essentially and produce carbon dioxide, which we breathe out, water, which we release, and then energy, ATP. So they're opposite of one another. So just as a recap or just as a disclaimer, please note that for AP biology, majority of the time, if you're given an FRQ or if you are given some sort of multiple choice question, they like to compare the two processes, photosynthesis with cellular respiration. So just to let you know, if you're reviewing for the exam, that you need to understand the differences between the two, especially if you're preparing for the official exam. And then if you are taking it in class, it's also really helpful to contrast both ways of production that occur between photosynthesis and cellular respiration because you could see that on exam. So know what goes in both steps and what comes out and how they work together. Now, to go back to this slide here, you'll see that we're talking about the difference between aerobic respiration and anaerobic respiration. Aerobic means with air, you can think of it, and anaerobic and meaning without air. What we mean by air, we essentially mean oxygen. So in aerobic respiration, we are utilizing oxygen, whereas in anaerobic respiration, we're not utilizing oxygen. For us as human beings, we participate in aerobic respiration. And the reason why we participate in aerobic respiration is because we need to produce a lot of energy to keep us going. And we also consume glucose as well. In contrast, anaerobic respiration does not produce a lot of energy. And we're going to talk about the different types of anaerobic respiration towards the end of this video. If you want to get more types of information related to anaerobic respiration, you can scroll down below where you can click the anaerobic related topics such as fermentation and understanding lactic acid fermentation versus the alcoholic fermentation. But for now, just know in the beginning of this video, we're focusing mostly on aerobic respiration, and anaerobic respiration is what we're going to be focusing on later on. So for now, you need to understand aerobic requires oxygen, which anaerobic does not. And the reason why we utilize aerobic respiration is because we need to release a lot of energy. Okay, so now we have our reaction, and we want to consider the fact that the understanding that cellular respiration is a redox reaction, which basically means that we're taking the glucose to go ahead and produce that carbon dioxide, losing electrons to oxygen. And that's called oxidation. Reduction is the process of taking that oxygen to go from oxygen to then go ahead and reduce, or I should say, reduce to form water to then gain electrons. So that's where the term oxidation reduction comes in, because we're taking our reactants, which are both oxygen and glucose, and we're either accepting or donating electrons to then become water and CO2. So just be aware of that when you're reviewing cellular respiration and understanding the chemical reaction in general. Okay, so now we're going to go into more details in understanding cellular respiration. Please note the following is a great cheat sheet to understand what goes in and what comes out on a high level scale. Please note this is not all the information that you need, but it's really helpful to really break it down when you're trying to understand cellular respiration. So let's take a look at the steps in general. So let's take a look at glycolysis, which is the first step that we're going to really dive into, which is the process of taking glucose and turning it into pyruvate. Specifically, we convert one glucose to then become two pyruvate. And the goal of this is to then have a net gain, and this is really important, a net gain of two ATPs. Now remember, glycolysis itself is a process, which means it's not just going to produce two ATPs, it's actually going to produce more, which we're going to get into more detail in just a second. However, the thing is with glycolysis, we need to keep glycolysis going, which does require some energy. So that's why we say it's a net gain of 2 ATPs. So just be aware of that. This process itself of glycolysis does not require any sort of oxygen itself, and so we consider that anaerobic. And this is important to note because it's the first step for respiration in both aerobic and anaerobic respiration. So just be aware of that. Also, please note in glycolysis, we're producing some NADPH, which will be used in the final step. Okay. Now let's go to the second step, which is the intermediate step. You don't really see this process listed in this picture here, but it covers this arrow here. And we're going to go into more details later on in the video. The goal of this intermediate step is to take this pyruvate and then transport it into the mitochondria itself. Remember, glycolysis is taking place in the cytosol, and that pyruvate is in the cytosol, but it needs to transfer over into the mitochondria. The only way that it could do that is by taking pyruvate and transforming it into what's called acetyl CoA. The acetyl CoA production makes the carbon dioxide or the CO2 release. So there is a CO2 that's released in this intermediate step. The acetyl CoA that's produced then leads to the Krebs cycle. So to recap, this intermediate step is allowing for pyruvate to go into the mitochondria by converting the pyruvate itself into what's called acetyl CoA. And that acetyl CoA then churns the Krebs cycle. Now, in some textbooks, they call the Krebs cycle the citric acid cycle. So just be aware that Krebs cycle can also be known as the citric acid cycle, and this occurs in the mitochondrial matrix, that inner membrane that is internal of the mitochondria. In this step, they produce some ATP, very small amount, and then you have some NADH that's also produced and the FADH that's also produced there. So NADH, FADH, and ATP. The CO2 is shown here, but remember that's from that intermediate step. It's already been released, right? So once it hits the Krebs cycle, it's gone. Now what's happening is we have that Krebs cycle that occurs, and the purpose, once again, is to take the ATP, produce a very small amount, and then have the NADH and the FADH that's produced here to then carry over to the final step. Now, in this final step, we're taking the energy that's being produced, essentially the NADH, and the FADH2 that's produced from both glycolysis and the Krebs cycle to really pump out that final step. In this final step, which is probably the most important step of breathing, is called the electron transfer chain. And it uses what's called oxidative phosphorylation, which we're going to cover in the later part of this video. It uses a process called chemiosmosis. And a brief overview is that chemiosmosis step is there to produce a lot of ATP. In fact, it produces about 32 ATPs every time that you are going through the step of cellular respiration. And when this occurs, you're producing all that energy and also releasing some water in the process. So that is a brief overview of cellular respiration. Now, in the next part of the video, we're going to get into more details about glycolysis, Krebs, and etc. So that way you can know more details about each individual step. When we're talking about the process of cellular respiration in general, we're talking about the ability that cellular respiration as a whole is able to produce energy, or ATP. Now, taking that information into context, I want you to travel back in time to the earlier part of Unit 3, in which you probably covered enzymes. If you haven't, make sure that you cover the earlier sections 3.1 to 3.5. There are videos in the Unit 3 playlist that you can review, and it talks about enzymes and how they formulate, and it gives you a better understanding, and it's great for understanding the first topic that we're going to be talking about. Now, when we're talking about the production of ATP or energy, there are two ways of formulating that ATP, and that's substrate-level phosphorylation or oxidative phosphorylation. With substrate-level phosphorylation, this is about taking an enzyme, and remember, we have an active site, and we have some sort of substrate that has to go ahead and attach, therefore eliciting some sort of reaction essentially. With substrate-level phosphorylation, what you're doing is you're taking an enzyme, in this case, it's pyruvate kinase. And what ends up happening is you're attaching it to what's called PEP. And what ends up happening is the process transfers the phosphate group. So this is a P attached to four O's. You're taking that phosphate group from that molecule of PEP, and you're going to take it and attach it to what's called adenosine, or AD, which is the same thing as saying that you have this nucleoside, which has this structure here, but I want you to focus in on those phosphates there. There's two phosphates. And if you think of ADP, you're thinking of two phosphates di, and you're going ahead and attaching another phosphate to that group. So essentially what happens in the reaction is you have pyruvate, which separates the phosphate and then goes and attaches it to that once ADP to become three phosphates. I know it's really tiny here, but it's three phosphates there. And what ends up happening is instead of becoming adenosine, it now becomes ATP. So the goal of substrate-level phosphorylation, to recap, is to take the enzyme, attach it to some sort of substrate, which allows for PEP, which has a phosphate, to transfer that phosphate over to adenosine, which has two phosphates originally, to eventually make three phosphates. So this process occurs in the first two steps of cellular respiration, which is glycolysis and Krebs cycle. So you need to understand that substrate level phosphorylation is very slow. And so because of this, it doesn't produce as much ATP. It's not as efficient, I want to say. Now, in the last step of cellular respiration, we're talking about what's called oxidative phosphorylation. What that is we're going to get into more details about it, but what ends up happening is you have a concentration gradient and you're producing all these hydrogens in these concentration gradients. And what ends up happening is it goes through this enzyme that's called ATP synthase. And the ATP synthase, I want you to think of like a treadmill. And it's taking all those hydrogens and it's taking the ADP and utilizing the hydrogens to then eventually produce ATP. And it does this like a chain, hence the name electron transfer chain. It's producing these electrons, hydrogen, and sending it through the ATP synthase to essentially take ADP and then attach it to an additional phosphate to become ATP. It's very efficient and it produces 90% of the ATP that's required for cellular respiration as a general process. So that's the difference between substrate level phosphorylation, which occurs in glycolysis and Krebs, which are the first two steps in the process of cellular respiration. However, the last step utilizes oxidative phosphorylation in the electron transfer chain, which is very efficient and very important, producing the most number of ATPs as required for cellular respiration. Now let's talk about the steps involved in glycolysis. The main objective of glycolysis is to take the glucose that we consume from our foods and go ahead and produce two pyruvate molecules. So note, for every one glucose molecule, you have two pyruvates that are produced. Now, the steps of glycolysis, there are 10 steps total, and they're categorized into the energy investment phase and the energy payoff phase. And the two steps together allow for the production of two ATPs, two NADHs, and two pyruvate, alongside with two H2Os, or two water molecules. This whole process of glycolysis in general occurs in the cytosol. And in this whole process, there really is no generation of O2 that is required, which means that as soon as we breathe in, we're not really using it for glycolysis. So therefore, we're considering this to be anaerobic, which means that for the step related to glycolysis, you are not requiring any oxygen. Now you see this chart here and it seems to be very complicated. And I tell students what you need to know is this net overall reaction because it helps you break it down. Now I'm going to go over the energy investment phase and energy payoff phase just so you can understand what's really happening here, but you don't need to memorize the intricacies of what goes in and out. So what happens in terms of glycolysis is you have some glucose that comes in and you have some ADP that's somewhere along the phase that also eventually creates two ATPs. And the ADP to ATP, the way that you want to think about it is the ADP to ATP phase, those two ATPs are going to be let go. And the reason why they're let go or they're let go of is because we need ATP, the two ATPs that are produced from these two ADPs, to go ahead and take the glucose to help it break off or break down. Remember, our end goal is to take this glucose and produce 2-pyruvate. And to do that, that requires some sort of energy. And that's what happens in glycolysis. You have some sort of energy that's already cyclical. Remember, glycolysis is occurring in the cytosol, in the cell. And in glycolysis, you're going to have some sort of energy that's required to help break down glucose. Now, when that is happening, you have some energy payoff. You have four ADPs that are somewhere in there that eventually produce four ATPs. You also have two NADHs that are there that eventually produce two NADHs. And at some point, you have some pyruvate that's produced, and that's from that glucose breakdown. So essentially, overall, what we're having is glucose, which produces our 2-pyruvate and water. That becomes the breakdown. And you have the ADP plus the phosphates that are there that end up producing a net gain of two ATPs and the two NADHs that somewhere fall in that reaction to eventually produce two NADHs. So overall, in this reaction that occurs in the glycolysis, you have one glucose molecule that comes in, and essentially what ends up happening is that one glucose molecule will produce the two ATPs, the two NADHs, two pyruvate, and the two H2Os. But glucose essentially's main goal is to produce 2-pyruvate, and the process itself is what's producing the ATP and the NADH. And the glucose breaks down pyruvate and also has a byproduct of water. So that's where that water comes in. It's not coming from internally, it's coming from the glucose that goes and breaks down to 2-pyruvate and has leftover molecules, which formulates water. So I hope that helps understanding a brief overview of glycolysis so you understand that glucose goes in and one glucose molecule turns into 2-pyruvate. This occurs in the cytosol as well. Also note in this whole process, we have no water production that is utilizing oxygen. So what I mean is even though we have oxygen that is produced in the water, we're not utilizing any oxygen itself. And because of that, it is considered anaerobic because we're not requiring any oxygen by itself. Okay, so for glycolysis, we're going to focus on the two main portions of glycolysis. Please note that you don't really need to know one specific molecule to the next specific molecule if you're talking about AP biology. For college biology, I would say you would sometimes need to know something like this. Just because they might have you take a class as it relates to biochemistry, which this is honing in on the beginning of biochemistry. So I just want you to know that you have to know the overall investment of what goes in and what goes out. This is just extra information so you understand where things are coming and going. So when we talk about glycolysis, we have the first series of steps, which is part of the energy investment phase. In this energy investment phase, we're taking glycolysis and eventually going down a chain of events to get to G3P. Throughout this whole process, we have this moment in time where we're taking ATP, specifically two steps in here, and utilizing the ATP from the external environment to then produce ADP. When we're going ahead and doing this twice, step one and step two, we see the two conversions. They're not necessarily step one and step two, but in two steps in the series, we're seeing this conversion of ATP to ADP, which essentially means that we're going from adenosine triphosphate, which has three phosphates, to adenosine diphosphate, which has two phosphates. So what ends up happening in the leftover process of the phosphates that sort of come off, well, they attach to these structures here to allow for the process to continue and allowing for one molecule to turn into the next, specifically allowing for these steps to happen where glucose eventually turns to what's called G3P. Now, We just covered this here, but what you need to know is this is called the energy investment phase because we're utilizing ATP, specifically taking one of the phosphates off to then produce the molecules listed here. Now, if we're talking about the energy payoff phase, which is the latter part of the two, in this portion, we're taking that G3P and eventually converting it to pyruvate. However, in this entire process, we're going back and forth, taking ATP and using up the phosphate or releasing the phosphate to eventually get to pyruvate. Because of this, we know that the energy payoff is the fact that overall we're utilizing 4 ATPs to eventually get to the 2 ATPs. And if you look at this entire reaction here, you could actually see the ATPs 1, 2, 3, 4. So that's important to note here. Now, if we remember, when we talked about in the beginning, glycolysis overall is about glucose, 2 ADPs, 2 PIs, and 2 NAD pluses that are going in, and 2 ATPs, 2 NADHs, 2 pyruvates, and 2 H2Os or water are output. So you do need to know that for both the energy investment phase and the energy payoff phase. But I just wanted to go over this just so you get a better understanding of what's really happening with those ATPs. Now let's talk about the intermediate phase. Now remember, glycolysis takes place where? In the cytosol. And at this moment, we've gone and created pyruvate. Remember, for every one glucose molecule, you have 2 pyruvate. And what ends up happening is before we go from pyruvate to going inside, from outside the cytosol to inside the mitochondria, there's a step. It's called the intermediate step. So we're going from the glycolysis step, what the products were produced, before going to the next step, which is called Krebs or citric acid cycle. We have this middle step, which is called the intermediate step. The intermediate step is the goal of taking cytosol, the location where this occurs, to then eventually go to the mitochondrial matrix. So I think the picture or the table here is really good at breaking down what we're going to talk about in just a second. But if you want to take a screenshot or understanding the location, which you are expected to know, and the description of what is occurring, you can do that here before we go over. So I want to also note that, remember, we have one glucose molecule from glycolysis, and that one glucose molecule conducts or creates 2 pyruvate. So when we're talking about this overall reaction, we're talking about the 2 pyruvate that's eventually going to actually produce double what you see here. We're only focusing on 1 pyruvate here. But imagine if we are talking about a full glycolysis reaction, this would be doubled. For the purposes of demonstration, we're just utilizing 1 pyruvate. So you have pyruvate, remember there's usually 2, that's made in the cytosol via glycolysis and is going to be eventually transported to the mitochondria. As it enters, what ends up happening is we have this barrier from the cytosol to the mitochondria. There are a bunch of things that come in and out. And one of those things from the pyruvate that we see as pyruvate enters is it loses carbon dioxide or loses carbon essentially from CO2. And then what ends up happening is, remember, that's when we breathe out. Remember, we breathe in oxygen, but we release CO2. And that's where that's coming from. We also have this idea that there is this CO2 that is coming up from 1 pyruvate. I just want you to remember there's 2 pyruvates, right? For one glucose molecule that goes through glycolysis, which means that this 2 is doubled. You're actually going to have 2 CO2s that are released, not just one. Okay, so in the second part of what's being released is this idea that we have the remaining 2 carbon fragments that are going to eventually form what's called acetyl CoA. So the goal is to go from pyruvate to acetyl CoA. However, when doing so, what happens is we have to ensure that we have some sort of coenzyme, noted as coenzyme A, that helps facilitate the reaction. Also, in this process, we're releasing some NAD to become NADH. So that's also something to note as well. Now, where the hydrogen comes from, that hydrogen is, remember, coming from the the ability to convert NAD to NADH and then have that hydrogen come in as well. So we have this idea that we're forming that acetyl CoA and that we're taking the NAD plus and formulating NADH by the addition of water, or I should say hydrogen essentially. Now remember, we have one glucose molecule that's going to double everything that we see here. So we not only have two CO2s and two pyruvates, we have the idea that we're also producing two NADHs and NADH plus, I should say. With that being said, now we're in the mitochondrial matrix as well in this transition, and we know that acetyl CoA will go ahead and formulate because of the coenzyme A, which helps us go to the next step. Once again, because in fact we have two pyruvate, this acetyl CoA would actually be two in reality, not one. Now we're going to talk about the Krebs cycle. The Krebs cycle is what you see on the left-hand side, but we're going to go over some big ticket items here. The first thing that you want to note is if you want to go through step by step and understanding what each step and what each molecule is, you could take a look at this picture here, or you can go ahead and take a look at the steps here. The Krebs cycle is going to produce one overall ATP, three NADHs, and one FADH2. The function of the Krebs cycle is basically to generate these electrons that you see here and go ahead and utilize that for the very last step of electron transfer chain. Now remember, in our preview, we said that the process of glycolysis and the process of Krebs cycle, also known as the citric acid cycle, is not going to be producing a lot of ATP. In fact, they produce very little. However, the reason why we have those ATPs is to basically help us go to the next step. We have what's called electron carriers, which are those NADHs and FADHs too. And you'll notice here they have hydrogens, which is important to note for the next step, but these all go into the electron transfer chain. There is, in fact, a total of 8 steps for the citric acid cycle that you are expected to know that we're going to go over in just a second. But you need to know the brief overall of what's happening to really understand what's happening in this cycle. Now, for one glucose molecule, remember, everything is doubled for the Krebs or citric cycle because remember, we're talking about one acetyl CoA, but that's not necessarily true because remember, we have one glucose molecule that produces two pyruvate, which also produces two acetyl CoA, which means that this cycle of the citric acid cycle will not just run once, but twice to compensate for the two acetyl CoAs that are coming out for the one pyruvate. This also means our NADHs and our FADHs and our ATPs are going to be doubled. Remember, it's now going through churning 2 times for the reaction to occur, which essentially means that we're producing 2 ATPs, 6 NADHs, and 2 FADHs. Important to note. Now, for CO2, please note that they're going to be released as well in this cycle. And when that occurs, remember, we're going to the cycle twice. So we're not just expecting one CO2 release, but 2, because you're going to have one glucose molecule that's coming in, and it's actually going to come out twice, I should say, for the one spin. But for 2 spins, that essentially means that there's 4 CO2s that's being released. So to recap for the CO2, usually we have 2 CO2s per citric acid cycle, but because we're spinning twice, we double that. So essentially we have 4 CO2s. Now remember, entry into the citric acid cycle basically means that we've converted in that intermediate step pyruvate to acetyl CoA and we've doubled it, which means that this process that we're seeing on the left-hand side here will be doubled for one glucose molecule. Each of the steps listed here also will be doubled. So I want you to note that when trying to remember Krebs or citric acid cycle. If you want to know the reactants and the products and what goes in and out, you can review the steps listed below. Okay, so now let's talk about the electron transfer chain. We now know that the electron transfer chain occurs in a phospholipid bilayer, which is actually not on a outside of the cell. When we're talking about comparing what it looks like, a phospholipid bilayer in this case is the phospholipid bilayer inside the cell, specifically on the mitochondria, and it occurs in the inner mitochondrial membrane. Remember, there's cristae that increase the surface area. The goal of the ETC is to utilize, remember, in the previous steps, we had NADHs and FADHs. And remember, I told you those two steps are important because what's happening is we have these hydrogens here. These hydrogens are somewhat important, actually very important, because the process is to take these protein processes to then go ahead and convert to have hydrogens to eventually produce our large quantity of ATP. And how this happens in the electron transfer chain is you have these proteins, also known as electron carriers, that are in steps. So this is step 1, this is step 2, this is step 3, and this is step 4. In the process of step 1, we're taking NADH that we had previously from the first and second part of cellular respiration, and we're removing the hydrogen to then pump into the electron carrier 1. Now, while this is doing simultaneously, you have a FADH2, which came from the previous first two steps as well, and releasing that hydrogen in the complex 2 to then go ahead and release FAD. So we have all these hydrogens from step 1 and step 2, and also step 3 as well. When I mean step, I mean protein 1, 2, and 3 that are pumping these hydrogens with the assistance of NADH and FADH, which will release their hydrogens into the inner membrane space. This continues on to the protein listed as 4 here. However, with that, we have some assistance. Remember, we consume water. Right. But we also release water as well. We have hydrogens and oxygen that we breathe in. So we have hydrogens, not that we breathe in, but hydrogens essentially that come from the loose hydrogens in this environment that go ahead alongside with oxygen to produce water. And this is where the water gets released in that step 4 process, along with the idea that the leftover hydrogens that are here are going to pump into this thing called the ATP synthase. Just to note, we've just covered the electron transfer chain, but we're also covering what's called chemiosmosis in just a second. However, we have all these hydrogens. They're going to be pumped into ATP synthase to eventually take ADP plus P to produce ATP. So this process of producing ATP through ATP synthase is called chemiosmosis and is important to note. Please note that all of these are the goal of step 1 and step 2. The idea of glycolysis and Krebs is to produce all these different electron carriers to then run the electron transfer chain to be very efficient in producing a lot of ATPs. Now, when we talk about ATP synthase or chemiosmosis, we're going to go into this idea that they're utilizing a concentration gradient. And that concentration gradient is going to allow for hydrogen to move in the protein. When this is happening, it's pumping hydrogen into the inner membrane space, and it establishes some sort of a gradient to allow ADP and that phosphate to combine together to produce ATP and some water as well. Just to note, the flow of electrons through ETC creates and assists in this gradient as well. So all the different 1, 2, 3, 4 that we talked about in the electron transfer chain is leading to this process here. Remember, NADH and FADH2 were delivered as electrons and had hydrogen on them. They're going in the first steps of the ETC and breaking that down to release the hydrogens. So what happens is those hydrogens get pumped outside into the inner membrane space where they establish this concentration gradient. The protons will then diffuse back into the matrix in what's called the ATP synthase right here, and the production is being able to produce all this ATP. Remember, it's very efficient. We're talking about oxidative phosphorylation, and we produce a substantial amount of ATP. Now let's go over anaerobic respiration. Anaerobic respiration is different than aerobic respiration. Anaerobic respiration does not use any oxygen, right? But aerobic respiration has its own benefits as well. It's very efficient. It allows the idea of producing ATP with very little efforts. And while it does use oxygen, the benefits of aerobic respiration is this idea that you can utilize oxygen to help you efficiently produce enough energy versus anaerobic respiration does not utilize oxygen and it actually takes a lot of effort to produce any form of energy in these two steps. Let's talk about fermentation more in detail. So when we're talking about fermentation, fermentation is the anaerobic conditions. Remember, we previously talked about cellular respiration and we're focusing previously on utilizing that oxygen. Remember, that oxygen is the idea of going from glycolysis to Krebs to etc. But what happens if we don't have oxygen once we reach that first initial glycolysis step? Remember, glycolysis itself is anaerobic because we don't utilize ATP. So for glycolysis and fermentation, what happens is they all go through that glycolysis step. But when they're in anaerobic conditions, meaning there's no ATP, they don't really further go into Krebs or etc. What ends up happening is there's two types of fermentation, alcoholic or lactic acid, which is utilized or anaerobic, which means that they don't use oxygen. So alcoholic fermentation is the first one, and then the other one is lactic acid fermentation. And they produce ATP pretty efficiently. I shouldn't say efficiently, pretty reasonable, but it doesn't produce enough. It's not as efficient as cellular respiration. So when we're talking about alcoholic fermentation, we're talking about this idea that we're taking some glucose, we're conducting some glycolysis, and we still produce the two ATPs. But what ends up happening is instead we produce ethanol by going ahead and converting NAD plus to NADH in the cyclic moment and also releasing some ATP. This is great for things like brewing, baking, and winemaking. In difference, we have lactic acid fermentation, and the goal of this is to produce ATP or NAD plus for glycolysis to generate some sort of ATP. Once again, not as much as an electron transfer chain, but still another way of producing some sort of energy. And an example of this is when pyruvate goes and converts into lactic acid. What ends up happening is you can take on this idea of having glucose, turning it into the 2-pyruvate, and then undergoing fermentation and converting each of the pyruvates into what's called lactose. Or if it's double, it's 2 lactoses. Because remember, essentially from one glucose molecule, you have 2 pyruvates, which eventually leads to 2 lactose. The lactate itself also has the idea of going from NADH to NAD plus and utilizing the hydrogens over and over again for the processes of fermentation. So when we're talking about glycolysis itself in fermentation and comparing it to cellular respiration, you could potentially see a FRQ that compares the steps of glycolysis, the steps of Krebs, the steps of etc., combines them together and compares that to anaerobic versus aerobic respiration. And the thing you need to understand is evidence does suggest that there's some sort of metabolic pathway that occurred very early on. What happened is in the very early forms, we did not rely on oxygen as a species or just in general for organisms. And what ended up happening is that you had some sort of oxygen. And at that time, there was this idea that the old-fashioned prokaryotes somehow utilized glycolysis and then produced ATP in that step. And remember, glycolysis does not require oxygen. But what ended up happening was then continued steps of Krebs and etc. were discovered to utilize oxygen themselves. And hence the 3 terms that you need to remember with this. So if you're talking about obligate aerobes here, you're talking about those organisms that require some sort of oxygen and perform respiration. Think about you and me. Obligate anaerobes cannot survive on oxygen and rely only on fermentation. So they'll depend on things like lactic acid fermentation or alcoholic fermentation. And then facilitative anaerobes are those that can survive with or without oxygen. They go back and forth. And they're great for environments that constantly shift back and forth. Now, ready for a quiz? Let's take a look at the following question here. Take a moment, pause the video, and then we're going to try to answer this question together. Okay, now that you're done reviewing the question and potentially answering it, let's take a look at this question together. It says some friends are trying to make bread in their kitchen and they've mixed yeast with dough and they let it sit for several hours. However, they notice that the dough has risen, but no alcohol has been produced. So what do we know about this? First of all, we know that if we're talking about bread making, which requires some yeast, and they say that there is this idea that it's risen, they're talking about an environment that doesn't have oxygen. Remember, yeast depends on some sort of alcoholic fermentation. Therefore, we can eliminate answer choice B, which says the mixture needs more water, and answer choice C. Now, we know that yeast is considered to produce alcohol when there is no oxygen, so answer choice D would not be the great answer. And the correct answer would be A, because we know that when we're utilizing yeast for anything, they don't not only need oxygen, they only need alcohol to survive. I hope things like this help this specific video for understanding cellular respiration. Make sure to hit that like button and make sure to subscribe, and we hope to see you back soon. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our Linktree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams. We can't